This is Mr Karthikeyan. Greetings to you all. Welcome back to our video session. In this video module, a unique topic I have brought for you, that is regarding viruses. We knew already what are viruses. Viruses term is derived from virion, poison. Virus in the sense of death. Remember, study of virus is called virology. Actually, virus is not a cell. Virus is a cellular organism. Outside of the host cell, it is inactive. When it enters into the host cell, it becomes active, because viruses are called obligatory parasites. First time, Ivanovsky virus existence is discovered. Then later, after Ivanovsky, Beijerinck, the scientist Beijerinck, he was noted and stated viruses as vivum fluidum. Vivum fluidum, nothing but poisonous fluid. So that poisonous fluid is responsible for the death or fatal of other organisms. So viruses, they can attack or affect all types of organisms on this earth. Some type of viruses, they affect plants called phytoviruses. Some type of viruses, they can affect animals called zooviruses or zoophages. Some type of viruses only they can attack bacterial cell called bacteriophages. So here bacteriophage schematic presentation we have on the board. This bacteriophage, again different types are there. T1, T2, T3, T4, T5, T6, T7. These all are bacteriophages types. These all belongs to the family Myoviridae and the subfamily T even Viridae. Among these all, one of the virus is known as T4 phase or bacteriophage or T even phase. We are going to discuss. Then who gave this information of viruses? Nothing but ICTV. Same like ICZN and ICBN. We have in our biology ICTV, International Committee on Taxonomy of Viruses. I repeat again, International Committee on Taxonomy of Viruses. So here, same like ICBN and ICZN, these hierarchy levels given by that committee. In viruses classification, first thing family, next subfamily, next genus, next the name of the virus, in the sense the species. So the species of the virus is, the name of the virus is given by based on the disease or the disease symptoms which the viruses cause. Okay, this information is on the other hand. In one more video I shall explain to you in depth. Coming to our main content, bacteriophage or T even phase. If we observe keenly, bacteriophage, it has the main parts like head region, collar, tail region and tail plate, tail pins, tail fibers. Once again I repeat, head region, collar, tail region, tail plate, tail pins, tail fibers. One by one I shall tell you. First, coming to head region. Head is polyhedral in structure. Different types of viruses, they have different types of shapes and structures. Rod shape we have. This is polyhedral shape. Peplous shape we have. Spherical shape we have. And what it is called? Helical shape we have. These are the different shapes of viruses. Then coming to here, this is polyhedral. The head is made up of proteins because this is called capsid. Here the protein head is called capsid. In some type of viruses, along with the protein, lipids and glycoproteins, carbohydrates also will be involved. Such type of viruses are on another hand. So here this is called the capsid, and the capsid is made by protein subunits. Those are called capsomers. How many capsomers are located or present in the sense? 298 capsomers together form this T4 phase head or capsid. Exactly inside the capsid, double-stranded DNA is there, that is known as genome or genetic material of this bacteriophage, because this is adenovirus. Why? Because DNA is as a genome. Okay, then the head is connected to tail with the help of short structure is called collar. Here sometimes collar fibers also can be observed. Collar fibers. Here we did not give, but collar fibers also found. Followed by tail. This tail is made up of or covered by tail sheath. An important point, every virus it has only one structure, helical or spherical or peplose. But coming to our T4 phase, head is polyhedral and tail part is helical. So here dual structure it has. Head is polyhedral and tail sheath arrangement is helical. Dual structure. Remember. Then at the end of the tail plate is located. To the tail plate, 6 sharp tail pins are found and 6 fine and sharp lengthy fibers are arranged. Those are called tail fibers. Mainly this bacteriophage attack E. coli bacteria, only E. coli bacteria, Escherichia coli bacteria. The shape of the T4 phase is a tadpole shape. Then with the help of fibers, it will land on the bacteria E. coli, and with the help of pins and by secreting special enzyme, lysozyme, viral enzyme, that enzyme it dissolves the cell wall and membranes of that particular E. coli bacteria, and it will inject this genome into the bacterial cell. Here in viruses, there is no reproduction process. That process is known as multiplication. That's not called as reproduction. So, for the multiplication purpose, this bacteriophage requires 37 degrees centigrade temperature and 30 minutes to complete one cycle. That is the time period and the temperature requirement. In one cycle, one multiplication, 100 or 150 progeny is synthesized and what it is called, assimilated, in the sense different parts are formed like a machine inside the bacterial host cell. After assembly, those all will be released out. That is called burst size. That condition is called. Here, viruses generally perform 2 types of cycles for their multiplication. One is a lytic cycle, second one is a lysogenic cycle. So the length is 200 nanometers and the width or diameter of this virus is 90 nanometers. That's it. Everyone, I hope you understood. Next video we shall meet with one more unique topic. This is Mr. Karthikeyan, mentor and expert and educator in competitive biological sciences classes, botany and zoology. Thank you. Signing off.